The first systemic antibacterial agent to be put in the market are the sulfa drugs, and particularly the first one to be developed is Prontocel. These drugs were developed for systemic use by Bayer Pharmaceutical in the 30s, and conceptually they started from the idea that compounds that would stain bacteria could also be used as magic bullet to kind of selectively target bacteria cells over the host cells. So the first drug is Prontocel, which is actually a diazo dye, and so it's a compound that's colored, as you can see by the heavy conjugation throughout this molecule. This is actually a prodrug, and so it's only active after it's metabolized the active compound, and the active compound is a sulfonamide. So there's a compound in which the aza bond has been broken up, and this is actually the active component. The structure activity relationship for this class of drug is pretty simple. So the only thing we can really change is R2. The pharmacophore includes the aromatic ring, includes para substitution. There cannot be any linker here, and the R1 group needs to be cleavable. So these drugs were discovered in the 30s, and they were pretty much the only antibacterial agents available before the advent of antibiotics, and in particular the discovery of penicillin. They are credited to save many lives during the Second World War, and particularly in the life of Winston Churchill, who got pneumonia in 1943. Other patients that basically helped publicize really the use of sulfa drugs. Was one of the sons of Franklin Roosevelt was treated with a sulfa drug for bacterial infections and survived. In general, we will use them as prodrugs. These compounds, in fact, can be optimized in terms of absorption, and particularly in the GI tract, by masking the amino group on the aromatic ring with a number of groups, and in this case an acetyl group is sufficient to improve absorption, and then the acetyl group is cleaved enzymatically after absorption in the GI tract. So realistically, all the analogs we'll see are actually the R2 group, and we can explore different aromatic and heteroaromatic rings at this position to improve activity. In terms of applications, those were, again, the antibacterial drugs of choice in the 30s, and they have been now superseded by penicillins and other antibiotics. However, they're still important today. One important thing about sulfa drugs is that they tend to concentrate in the kidney, and that can be a problem because they tend to precipitate and therefore damage the kidney. But it can also be used in therapy because they are useful for the treatment of urinary tract infections. They're also used topically in eye lotion, so to treat eye infections. They can be used in the treatment of GI tract infections, and in this case they are not absorbed, and they serve to basically kill noxious bacteria in the GI tract. They're not very selective, so they will tend to kill pretty much anything in the GI tract, and so as many antibiotics, one of the problems can be then the bacterial flora can be a little slow to restore after treatment. And they're also used in treatment of mucosal infections of various types. So what is the mechanism of actions for these drugs? Well, so these belong to the antimetabolite class of antibacterial agents, and in particular they are involved in the synthesis of tetrahydrofolate, which is coenzyme F. And both bacteria and humans need tetrahydrofolate to function, and we'll see how they're basically involved in the synthesis of DNA bases. However, humans have a transporters, and so we actually take folic acid from the diet. We do not make our own. Bacteria, on the other hand, they lack the transporter that can take up folic acid, and so they have to make their own. And the way they make them own is to make dihydropteroate using para-aminobenzoic acid and dihydropteroate synthesis on this substrate, which is the diphosphor form. And sulfa drugs serve as a reversible inhibitor of this enzyme. Once the dihydropteroate is formed, it can then be functionalized with glutamic acid to form dihydrofolate, which is then reduced in the presence of an NADPH to tetrahydrofolate. And we can inhibit this pathway in bacteria at two levels. Dihydropteroate synthase is inhibited by para-aminobenzoic acid, and dihydrofolate reductase is inhibited by trimethylprem, which we'll see in a few slides. What does a folic acid do for us in the cell? Well, as I said, they're involved in the synthesis of DNA bases, and particularly of pyrimidines. And because they block this synthesis, they are considered bacteriostatics. In other words, they don't they don't kill the bacteria, but they rather prevent their growth and multiplication. And in particular, they're active on thymidylate synthase, and so they transform DEUMP into DTMP, and they do that by transfer a methyl group here at this site. Other parts where the other metabolism where they're involved is in the synthesis of formyl-methionine tRNA, so the tRNA that carries methionine. They're involved in the synthesis of serine from glycine and methionine from homocysteine. The mechanism of action. Once again, they're competitive inhibitors, so they bind to dihydropteroate synthase in the same place where PABA binds, para-aminobenzoic acid binds. And as you can see, that's the reason why only para substitution is allowed, why the aromatic ring is necessary, and why we can't really touch either the amino group or the sulfonamide group too much. And so only one substitution on the R group is possible. They are metabolized in vivo by N-acetylation, so the reverse of the reaction we just saw in terms of making prodrugs, and this increases the hydrophobic character, which is good for absorption in the GI tract, but it's very bad in terms of reducing aqueous solubility and can lead to precipitation in the kidney, causing kidney damage. So how do we reduce toxicity? Well, we do that by increasing the solubility and electronic properties of the sulfonamide by using substituents at the R2 group rather. So let's look at a few examples of these sulfa drugs. Sulfadoxine is a new generation, so-called, because it has long-lasting antibacterial activity. It's it has a long half-life, so we can dose it very once a week. As many of the sulfa drugs is actually taken up together with something else, and can also be used in the treatment of malaria. As we will see, for the treatment of malaria in particular, we tend to mix up the drugs used because we want to minimize side effects, and also we want to minimize the insurgence of resistance. Malaria is very susceptible to resistance, and so we'd like to use two different drugs to hit the plasmodium falciparum at 2 different levels. Other compounds that are currently used on the market are, prodrugs, and so, succinyl sulfathiazole is one of them, and so as we can see, this group then gets enzymatically cleaved to succinic acid and releases the sulfathiazole, which is the active drug. Other types of prodrugs contain the benzoyl group here, and so they're cleaved into benzoic acid and sulfamides.